heart rate variability code has been around in my mind for some time. And so I would say if I were to try to define what do I mean, I just went straight to the Webster Dictionary, or forgive me, I think Oxford Dictionary would say the same thing, I presume. And it is that a code should be a system of signals or symbols that are used for communication. So can we identify features of the properties of the heart rate variability that would satisfy the criteria for something that we can call a code? And why should we care? I hope that these two things should come out. And I will attempt to demonstrate it to you by covering the properties of a time structure of heart rate variability, state space, and specificity and universality of this code. And I will go through examples in physiological experiments, mostly in fetal sheep that you now know extremely well through the previous talks, and as well in the clinical studies. So first, looking at the time, at the features of dependency and time resolution, the sampling rate offers itself as a nice way to gauge that. And I'll show you some examples looking at studies of fetal asphyxia and the conditions of umbilical cord occlusions, again, that you've seen already a few talks on, and conditions where cardiovascular decompensation occurs. You'll see in a moment what I mean. So last time, 2 years ago, I had the honor and the pleasure to show you our work on fetal EEG. And this time I'll focus on the cardiovascular features and the heart rate variability. In fact, in the same experiments that you just saw again a few talks on. It's been a treasure trove of data. You see here a recording in a chronically instrumented near-term fetal sheep with electroencephalogram, electrocardiogram, arterial blood pressure, fetal heart rate, and umbilical cord occlusion pressure. So when the bars go up, like so, that means there is an occlusion happening. And so there is a period of about 4 hours of such occlusions, a minute apart, and at some point the occlusions become what we call severe, such that the blood pressure here, instead of going up as it should under conditions of a fetal heart rate deceleration induced by umbilical cord occlusion, the blood pressure in fact begins to decrease. So this cardiovascular decompensation, failure of the cardiovascular system of the fetus to maintain the cardiac output under conditions of occlusions, is interestingly occurring at different time points in each animal. And it approximately occurs at the pH of 7.2, but we found it really hard to say why is there something like a oxygenation status at baseline before occlusions or any other features that can explain when they begin to decompensate. So the question was born inspired by a basic physiological idea called the Betzel-Tiarish reflex. And that's a reflex that's been described over 100 years ago. It's a vagally mediated depressor reflex of cardiac output where the heart attempts to preserve its metabolic activity by decreasing the cardiac output proactively through the vagus nerve. So without that, since it's a vagally mediated response, we should see a change in a heart rate variability feature such as RMSSD, Rhythm sclera, successive differences of our intervals is kind of the the house pet of looking at the changes in vagal activity. And so with that, can we analyze the time course of RMSSD dynamics throughout the occlusion series and capture a moment when we suddenly see the hypotensive response? So this is the time course of pH averaged over the group of all animals. And so you see the decrease of pH with advancing acidemia, as you've seen before in the talks. And this is a change point analysis that we developed that attempts to predict the time when the cardiovascular decompensation occurs, essentially exploring the higher moments of the heart rate variability, the variability of the heart rate variability. So what's interesting here, the blue line is the RMSSD that was computed from the 1,000 hertz acquired ECG signal, essentially equivalent of TAF ECG, the transabdominal signal. And the red line is RMSSD computed with a precision of 4 Hertz, so as if you would have it if you use the ultrasound. And so it seems that, as we've seen before in a cruder way, that the change point algorithm seems to fail to predict the correct, basically time, the cardiovascular decompensation when you have the lower sampled signal. And this seems to be explained by the variability introduced by overestimation of RMSSD already at the baseline at the lower sampling rate. So takeaway one was that we can detect cardiovascular decompensation from the heart rate analysis, and point two was that it seems to depend on sampling rate, suggesting there is a time structure in the signal. And we were able to confirm the observation in clinical studies with our friends at the University of Milan and at York University in Toronto, using signals from TA FSCG and comparing it to the about 550 recordings from the Prague data set that, again, you all know very well. And so this is the root mean square error. You see the nicely the distribution here that is really tight. It's the prediction of pH at birth from 2 hours of fetal heart rate before the second stage of labor. And so the prediction is considerably better when the underlying data is extracted from the TAFSG than from the ultrasound, from the CTG, despite the fact that the data set here is nearly 5 times as big. So that's by evidence to support the notion there is a time feature, a structure in heart rate variability that depends on the resolution of time through the analysis of the heart rate variability. There is a dimensionality to the heart rate variability. That's now going to the space, to the state space aspects of heart rate variability. I'll go over a few examples in studying fetal inflammatory response and hypoxia, as well as the effect of the vagotomy on the heart rate variability features landscape. So first, a quick reminder, the inflammatory response of the body is controlled through the vagus nerve. We call it a cholinergic anti-inflammatory pathway. It's been described extensively over the last decade, or 2 actually decades now. So the afferent endings of the vagus nerve are capable of sensing cytokines, the molecules that signal injury, endogenous or exogenous, and there's a processing network in the brain that converts that information into efferent output, which signals to macrophages, among other cells, through a very specific cellular pathway to suppress the production of pro-inflammatory cytokines. So it's a reflex akin to the bioreceptor reflex, except it controls the inflammatory, basically the milieu interior of inflammation in the body. So fortuitously for us, the vagus nerve also innervates the sinus node of the heart, where we are capable of eavesdropping on that communication through analysis of heart rate variability. So we use that insight derived from studies in adult species to try to devise a heart rate variability index from a set of data in fetal sheep, where we injected the lipopolysaccharide, so it's the antigen expressed on the gram-negative bacteria, often used to induce inflammatory response, in an animal model and in humans, interestingly, if they agree, to measure the correlation between a subset of heart rate variability features and the rise of pro-inflammatory cytokine IL-6. I apologize for the size of the figure, but it's more the gestalt that I care about in this particular case. This is an axis of 54 hours. The LPS is injected here. IL-6 goes up within about 3 hours, and the heart rate variability index that we were able to derive looking at over 51 HRV measures and conducting some dimensionality reduction magic on them seems to follow exactly in time the cytokine expression and the profile that we measure in the fetal blood. So you have yourself fetal inflammatory index from the heart rate variability. And this is the systemic inflammatory response, a signature. Can we take that index and use it in a different animal model? We injected a sublethal dose of LPS to a piglet, a neonatal piglet, a great model for neonatal sepsis, and suboptimally shown agreeably. But in red, hopefully you can see this is TNF-alpha measured in the blood of the piglet, and this is only about 1.5 hours because it's a sublethal dose, so the piglet goes in a sepsis within about 2 hours, and it would die without ICU level support. And here is the increase of TNF-alpha within about 1.5 hours, and here is the heart rate variability inflammatory index that we inferred from experiments in fetal sheep, applied as is the heart rate variability that was acquired in the piglet, and it follows in time exactly the profile of the TNF-alpha increase. If we use the vagus nerve stimulation on a different animal exposed again to the same dose of LPS, we are suppressing the TNF-alpha expression, and we are also seeing the concomitant decrease in the heart rate variability index. So there seems to be a degree of universality to such an index from species to species. And I will not go so much into the vagus electroneurogram analysis, which is an area of interest to better understand the underlying physiology. So we also asked, can we see the correlation of heart rate variability features at the same time to inflammation in the organs? So we look at the gut. The terminal ileum is a locus minoris resistentia for necrotizing enterocolitis of the neonate. So it's a weak spot where you mostly see necrotizing enterocolitis in the neonate. Until now, we don't know which of the babies are going to get it. So there is an interest to identify babies who might be at the risk of developing NEC. Turns out there is a subset of features of heart rate variability that correlates specifically to inflammation in the gut and do not overlap with the heart rate variability features that describe the systemic inflammatory response or that describe the brain inflammatory response. So there seems to be an organ, a signature, or a target specificity of that purported code. Is there a memory of exposures in utero that the toddler or a neonate might be carrying with itself, herself, himself, that is embedded in the heart rate variability, kind of akin to epigenetics, how you think about. So we conducted a study using a data set of the Langendorf preparation at the University of Cambridge Dina Geoscience Lab that exposes the instrumented sheep in utero to the environment of the hypoxia for a month or so, and then after the necropsy, the heart is extracted, and the Langendorf preparation allows you to study the heart separated from the body ex vivo. It's a standard approach to study the physiology of heart ex vivo. We asked whether we can see a heart rate variability features that are autochtonic or intrinsic to the heart itself without any innovation, and that is the case. And interestingly, there seems to be evidence of a memory of heart rate variability in animals that were exposed to hypoxia in utero compared to those that were not. We then had opportunity to go to Porto Alegre a few years ago and got heart rate variability data from the toddlers born to moms that were diagnosed with a Zika virus infection but didn't have a symptom, and the babies are not showing any signs of a Zika infection like microcephaly. However, their HRV signature is distinct from those that do not have Zika virus. And interestingly, the features of heart rate variability signatures that distinguish them are from the same signal analytical domain as the features that describe the memory of hypoxia in utero in the sheep model. That is interesting because Zika virus is known to cause hypoxia due to alteration in the placental morphology. There is also observations that heart rate variability can distinguish kids, 8-year-old kids with autism spectrum disorder at the age of 8 from other kids. This is just the data showing the intrinsic HRV, and you can find this all online. And the relationship of this intrinsic HRV features to left ventricular and diastolic pressure, which is of interest, I think. So, and this is showing the ability to use heart rate variability features. This is worked together with Etienne, who is here. So we're actually able to identify fairly well kids who have depression from kids who have the conduct disorder from kids who have autism spectrum disorder. That's a group of 66 kids here. The question, of course, is if there are indeed such signatures that are also specific to conditions like these, can we go back in time and try to look for them after birth or even in utero to predict the alteration of developmental trajectories? This is where the monitor, such as the one that was just shown, might come in really handy. So there's also effects of the vagotomy on heart rate variability signatures, and again, they're not simple in a sense that only one or two are changed. The experimental design here was again in sheep. There was a surgery. Right in surgery, we installed the fetal ECG to record the fetal ECG throughout the surgery, and we had data before and after the vagotomy was done on 2 sides of the neck, so complete abolition of innervation for the vagus nerve. And so we looked at changes in heart rate variability acutely before, after, and 72 hours after the surgery. Interestingly, the heart rate doesn't actually change. If you cut your vagus nerve now, you would be very tachycardic. But in the fetus, that isn't the case. And don't cut your vagus nerve. But I was not recommending it. But interestingly, there is a distinctive set of changes in heart rate variability features, and we went through the entire zoo of those that are available to us. This is a set of about 51 heart rate variability measures, similar to those you've just seen that we use for derivation of inflammatory index. And there is 4 HRV measures that are distinctly changed due to the vagus nerve removal from the picture. So there is something akin of a landscape change that occurs, but it's not one measure, and it's interestingly not RMSSD, which kind of everybody uses, including me, as you've just seen. But it is all I'm trying to say here is not go through them, but to say there is a complex landscape of patterns of heart rate variability features that go up and down as a result of a surgery stress, a homeokinetic stress, if you will, or due to vagotomy. So really, to summarize my pitch for the existence of the HRV code, I think that HRV shows a rich spatiotemporal basically life or structure. The properties of heart rate variability seem to be state space and also time resolution dependent, as we've seen in examples of the influence of sampling rate on the estimates of some outcomes. And subsets of the heart rate variability measure seem to relate to specific physiological responses and are indeed akin to a code. I think that has implications for thinking about the data acquisition, depending on which outcomes you're looking to link it to, and for interpretation and biomarker discovery down the road. And if you're interested in what I mean with bioelectronic medicine, we can talk about it. I have to acknowledge a large team of people across the globe that I've enjoyed to work with over the years to get all this data. It's about 10 years worth of data, I think, and a good deal of support to get there. On this note, thank you. And
we have the time for some questions.
Yes, just a very short question, very interesting. My question is about the specificity of this kind of parameter, because in different contexts you can find heart rate variability indexes or correlating with several neurological problems and so on. So, for instance, you cited autism. It seems to me not so much specific. So, can you comment on that?
Sure. Autism, first of all, we call it ASD, the DSM for the reason that it's actually subsumming a spectrum of symptomatology. By the side of that, it's well described that autism is associated with changes in autonomic in ANS and the autonomic nervous system and in HRV. In general, what has not been done before is to attempt to take next to each other kids, or for that matter, it could be adults with autism and some other psychiatric conditions, and indeed ask the question, is it really specific? This is exactly what we've done. And I actually just got a note from a Journal of Autism and Developmental Disorders about half an hour ago that it's newly accepted. They want to change something, but so hopefully we'll see it. But yeah, the cool thing, the exciting thing is that there seems to be a good deal of specificity, and the machine learning models help to streamline it. My vision, of course, for these things is to really make them available as APIs so that you can take that and collect your ECG data and throw it at it and test it. This is really, if anything, I think an important call for all these models, derivation of high variability features. We should be attempting to create APIs so that other people can actually use them. Otherwise, I think we're getting better, but I think it would accelerate the progress quite a bit. So yeah, short answer. I think there's evidence for specificity based on that.
Yeah, that would be my question also, because we've seen many times in various aspects of medical care correlations, things that tend to be more frequent. But we're really looking in medicine for discriminative power.
Mm-hmm.
So it's specificity, yes, it's also sensitivity. And usually when we start off, we get very typical cases of diseases. But of course, as with all biological features, it's not just black or white, there's a gray. So I think that's where we lose some of the specificity and some of the sensitivity. Would you like to comment on that?
I could. I don't think I'd like it. There's not much to say, really. This is the reality of extrapolating. This is a limited data set, as every data set is. You have 66 kids, which in itself is actually a larger cohort, relatively speaking, in the literature. Not the largest, but one of the bigger ones, to try to juxtapose the different conditions. But, time will show how much it scales if you take external data sets to validate it. It's so far all we can say is we try to be as thorough as we can using the validation split and fivefold validation and all these things. But at the end of the day, one thing that I should note about this particular cohort, they tended to be on the higher end of IQ, which was one of the critique points of the reviewers. So this is the cohort we got. May that be a factor down the road? We don't know. I don't think it's known how much IQ influences HRV in a setting of autism spectrum disorder. I mean, you see how it gets really so multidimensional. The best you can do is try to establish something that can be tested in other settings. I feel that would be my short answer.
Yeah, sure. Okay.
Thank you for excellent presentations. My question is that you mentioned the intrinsic origins of the heart rate variabilities as Langendorf experiment from the hypoxia rules. If it exists, can we differentiate the intrinsic or the non-intrinsic peripheral variability in the clinically?
So I think we might. This is a very political answer, I know, but I'm getting there. But so the whole idea of HRV code sort of makes this implicit assumption that this is what we should be able to do. But I think the important take-home message is this is not going to be one feature or two features. This is going to be a landscape of features, or in some high-dimensional space that you like, where those features evolve over time and represent an entity, a phenotype. And that phenotype is what you're comparing. You're not comparing linearly just a bunch of features that are thrown together. So what you're seeing here is what appears to be a subset of features that have not really been used much in a context of the fetal heart rate variability. In fact, even in the context of adult HRV studies, but they have in common a number of interesting mathematical things that, maybe we have a discussion in the paper where we try to relate it to pacemaker synchronization and how it's known that the pacemaker cells that comprise this sinus node are not really all basically showing a heart rate of, let's say, 70. They actually oscillate at individual basically heart rate, and the heart rate that you see as a sinus node heart rate is an emergent phenomenon. There's a Nature paper by Dr. Glass from McGill from about 30 years ago that showed that. There's a beautiful application of chaos theory. And so, because it's a synchronization phenomenon, it's also subject to dependence on exposures to things like acidemia or LPS. And so there is some physiology attempting to show that it could be that intercalating LPS molecules might be altering the coupling dynamics, or pH decrease might be altering coupling dynamics due to metabolic supply issues. So you can go quite far trying to relate synchronization dynamics to some specific HRV features that might be capturing it. Of course, we went the other way around. We discovered the features and then tried to explain it, but it kind of makes some sense. So I hope it could be specific. That's basically subject to future studies.
Okay, thank you very much, Martin. I think we have to go on